I'm a developmental neurobiologist. I am interested how brain is developed, and in particularly the most human part of that brain, cerebral cortex. And as you know, if we are different from any other species, and if you find one organ that's responsible for that difference, is cerebral cortex. So it's very interesting to look how it develops, and it's related to, of course, stem cells or neural stem cells, because embryonic stem cells are now buzzword. Everybody is excited about that. But I'm not working on embryonic stem cells. I'm working on neural stem cells. And these cells are next stage. They are more committed, differentiated, and they are dedicated to make our brain. So, the secret about how our brain and cerebral cortex develop and how we evolved as a species during evolution is in the way how our cerebral cortex, or brain, is made from simple progenitors, cells which divide. We call it neural stem cells. They come from embryonic stem cells and then become more differentiated. And they, by sequential expression of genes and cell interaction, finally make that complex organ. And I am excited about that prospects of studying neural stem cells, but I can tell you that if, and some people recognize, it's difficult even to use embryonic cell for organs like a liver or heart. It is even more difficult to use it for repair of the human brain. It is not impossible, because word impossible doesn't exist in modern science, but it is difficult, yes, and we have to work on that. Now, why I say is more difficult in the brain or cortex, for that matter, than in other structures, and if you ask me just that I describe it in one sentence, I would say that unlike in other organ, function of each individual cell, neuron, depend on its position. And heart cells are doing similar thing, whether it's they contract, whether it's a left part or right ventricle in the liver, whether it's a left or right lobe in the liver, they basically do same thing. In the brain, every individual cell has assignment, and they come to that position, and this is what we are studying. How neural stem cells, which are producing neurons, form that complex architecture of our brain. And one interesting thing, none of the neurons in cerebral cortex are generated in neurons themselves. They migrate. So what we are looking is what controls production of cells, what control their differentiation to different types, and how they migrate to come at the proper address in that complex brain. It's a mind-boggling problem. But we have to solve that problem, not only because it's genuinely interesting and intellectually appealing, because we have to understand how they do it in the first place if we want to replace them. We cannot just take neural stem cells, inject, and hope for the best. We have to understand the steps that have to be involved in their incorporation into circuitry of the working brain, and that is a formidable task. Now, give you example how this cerebral cortex, convoluted, large structure, is formed. And here you see on my screen, we are studying molecular and cellular mechanisms of proliferation, of division of neural stem cells, their differentiation of different type, and their migration to the proper positions. And that's a very complex and research we obtained, but I here have a kind of animation which indicate to you the complexity of that and timing of sequential events that have to be achieved to form that, say, cerebral cortex. And see, we start here, you see, we have cells are generated, ventricular zone, this is where neural stem cells are formed, and then another type is formed, so-called subventricular zone, above that. They produce new neurons, which then migrate increasingly long distance to cerebral cortex in a very interesting way, by passing previously generated cells. And they have both aerial and laminar specificity. So they become of specific type. They have addresses where to go in the brain. They migrate along specific pathways, along specialized transient population of cells. And here we have timing. This is done on the basis of long research with many colleagues of mine, now professors and associate professors, but at one point postdoctoral fellow and graduate students. And cells, we label them, we see how they divide and how they migrate, achieve their proper position in the cortex. And you see here's timing in monkey, post-conceptual days. We label their DNA, specific marker for DNA replication, one of the nucleotides of the DNA. So it can be permanently labeled because it's tagged with radioactivity. And then we look where they achieve their position. And it's interesting, they migrate, bypass each one, and stop in the proper position. Why I'm emphasizing that? You see, we have mutant, genetic mutant, in which, due to single gene mutation, cells fail to migrate properly into position. And if cells is late just one day and end up in the wrong place, mouse reel. This is called a reeler mouse. That means that this mouse doesn't have properly functional cerebral cortex. And this is where we are looking whether during human development there are genetic or environmental factors which interfere with neuronal migration, interfere with their ability to acquire proper final destination, final address. And from there we can discuss pathogenesis of different disorders of higher brain function. But when you look how they migrate, and I can show you a movie, a real movie, this is animation of how cells migrate, and I can give you that. By the way, you could then put it there if you wish.